The Gram stain is a type of differential stain, meaning that it uses two different stains or two different dyes. The Gram stain itself is probably the #1 most important stain used in microbiology. What happens is you do the stain to help you identify the organism you have. What it will do is, for bacterial classification, it will divide them as either a gram-positive or a gram-negative. And the difference between the gram-positive and gram-negative has to do with the structural components of the cell wall. You do this staining process and they react differently because of that cell wall structure. And because you're using two different dyes, one will look one color and the other will look a different color. So you can differentiate between a gram-positive or a gram-negative. You can tell the difference between them. Besides being able to tell the difference between a gram-positive and a gram-negative, you can also determine the cell size. You can determine the cell arrangement and the cell morphology. Because you're using two different dyes or two different stains, we usually call the first one that you're going to add the primary stain, and the second one that you add will be called either the secondary stain or the counter stain. You can use either term for that. Like I said, this is by far the most fundamental stain done in microbiology. So if you're working in a lab, you need to know how this is done. As with most stains, you would have taken your sample, you would have made a smear on a slide, microscope slide, you would have heat fixed it already. Those you assume are done first. To perform the actual Gram stain, then once you have heat fixed your sample, then what you will do, first thing is you will add that primary stain, which is crystal violet. You can either put a drop on the area where you know you did the smear, or you can flood the stain. You will leave that dye, crystal violet dye, on for 1 minute. Then you will rinse it with water. Then you don't dry it. So on that wet slide, now you're going to add grams iodine for 1 minute. You then will rinse with water. Now you will add ethyl alcohol. There's two different ways you can do this. Personally, the way I was trained, the way I prefer to do it is I add the ethyl alcohol, leave it on for 30 seconds, and then I rinse it off. Some people will add the alcohol for 15 seconds, rinse it off, add the ethyl alcohol a second time for 15 seconds, and then rinse it off. And then a third way you can do it is just add the alcohol. You kind of hold the slide at a slant, and you just add enough alcohol until it runs clear, until you don't see any of the purple crystal violet running off. Whichever method you use, you're going to rinse it, and then you're going to add saffron. This is a red dye. You add that on for 1 minute, and then you rinse it off with water, and then you're going to blot dry and then view it under the microscope. Now, once you're viewing it under the microscope, a gram-positive bacteria will appear purple. It keeps that primary stain. The gram-negative organism will remain red. Why are they different colors? Because it has to do with the difference, once again, in the composition of that cell wall.